uh, mention to all the students here and all the participants that I have designed the slides in such a way that we will first discuss some basics of what exactly xenobiotics are and how microbes deal with those and how they are helpful to us. I mean, these microbes are helpful to us. And then gradually we will move toward what research demands in this particular aspect, this particular domain. And then I will share some of my research experiences that I have been doing over since say last 20 years, as more than 20 years I would say, beginning as a PhD student at Bose Institute, which is actually a DST institute in Kolkata, and then moving to University of Tokyo as a JSPS postdoctoral fellow, and then continuing as a faculty over there, and then coming back. So all the story I would like to summarize here in terms of certain research results that we have received over certain years. So my topic, the title, as you all know, is role of microbes uh, in protecting the nature against xenobiotics. So the most important word here is xenobiotic, especially for me because I have been dealing with this over last many years. So yeah, giving a little introduction about myself, not the professional introduction regarding what I do. This is what my family thinks, that I am a scientist. What my friends actually think about me is I'm a crazy scientist who looks something like this. What my boss or previously my supervisors used to think, that I'm just sleeping, doing nothing. And this is basically what I do. So if you are surprised by the look of this person, yeah, we all know that
Very good.
this is a janitor and cleaning I mean cleaning stuff that we see around us. So basically me as a researcher is nothing different than him, because he also cleans up the mess that is visible to us, using his tools. And these are his tools that you can see here, the broom and all. And for researchers like me, the tools are something else that we will discuss about. But I'm also trying to clean up the mess that we human beings have created around us. So talking about the mess or the pollution, we all know it's nothing new for any of you, as you can see in the images here also. So technically pollution is defined as the contamination of both physical and biological components of this planet, right? So the contamination can be any type. It can be heat, sound energy, radioactivity, or whatever. So any kind of contamination, whenever we are adding to the planet, that can be defined as a pollution. So now we are polluting the environment in several ways, but not all the ways are harmful for the planet. For example, as you can see here, I've classified the images as two different images. One is this, the vegetable remains, or the kitchen waste that we throw around every day, which over a certain period of time get degraded in the environment and it perishes from the environments. So these are not something that we are very much concerned about. Yeah, it creates a lot of smell. It fills up the land. That is fine, but still, it is not as harmful as the other one. That is the non-biodegradable ones, which persist in the environment since for say ages, some 100 years, sometimes more than hundreds, few more 100 years, as the plastics are very infamous for that. And where did it start from? It started somewhere in the earlier 18th century during the Industrial Revolution, because from there the pollution, the anthropogenic pollution or man-made pollution came into existence, and we have been doing so since so many years so far. So, for example, it is an old data, but still a valuable data. For example, in 1990, there were around 65,000 different chemicals in the marketplace. And you can imagine 1990 and so many years have passed now, how where this count might have gone so far. So, and most of these chemicals that we are talking about, this particular count, are non-degradable by living organisms, and they remain in the ecosystem. They remain as persistent chemicals for several years, and they are really toxic and something that are painful for us. Talking about the sources of such pollution, this is also not new for us, that chemical and pharmaceutical industries, they contribute a lot to this kind of xenobiotics, like plasticizers, synthetic polymers, so on, different aromatics, polyaromatics. And then again, pulp and like industries like pulp and paper bleaching industries, which are contaminating the environment with chlorinated compounds, organic compounds mostly. These are again a very priority pollutants. Agricultural practices or malpractices, I would say, where we have been using a lot of synthetic fertilizers, pesticides, herbicides, the chemical, toxic chemical ones. So many of them have already been banned, but again, we all know that um, illegally or legally, a lot of countries and a lot of farmers are still using these chemicals, these toxic chemicals, which is again ending up reaching the water, this water bodies or landfills wherever. And then again, the fossil fuels, the incomplete combustion of which these carbon containing fuels such as wood, coal, diesel, and other petroleum products again end up into a lot of different polyaromatics or polycyclic aromatic hydrocarbons that we will talk about. And again, these are the compounds which uh, you know uh, contaminate the environment and create toxicity for us, for different life forms, not only human beings. When I say us, means all the living forms present on this planet, whether terrestrial or aquatic. Now, talking about types of environmental pollution, you have all, or almost all of us have studied these things, read these things in the textbooks in schools or maybe high school and college. But still, I would like to recapitulate a little bit. We'll browse through, because without talking about these sources of pollution, we actually shouldn't move ahead, I feel. So air pollution, for example, sulfur dioxide, nitrogen oxides, carbon oxides, ozone, VOx, or what we know as volatile organic compounds, and these and other airborne particles, along with radioactive pollutants, are probably the most destructive ones that we can talk about. Adding to these are the motor vehicle emissions, that is the exhausts, the automobile exhausts, which are severely polluting the environment. And these images, we are not, I mean, we are very much aware of these kind of images, right? So, and we have learned about, acid rains and different toxic fumes and so many things over the years, that air pollution is indeed something uh, which we all are facing with, especially talking about India. We know that cities like Delhi and Haryana, how much they are struggling these days with the smokes and smogs, whatever we call them. And then comes water pollution. And thanks to the a lot of industries that we can see here, which are uh, very freely and very happily, they are ejecting or um, dumping these wastes, these chemicals, sometimes radioactive contaminations as well, indiscriminately into the environment, into the water bodies, and thus polluting the water bodies. For example, different factories, refineries, tanneries, waste treatment plants, so many industries are there which are practically doing this every day. There are certain so these are the direct sources that I was talking about. There are certain indirect sources as well. For example, the agricultural practices, like whenever we are adding fertilizers or pesticides into the soil, uh, they uh, again end up reaching the this water bodies. So directly, indirectly also we are contaminating. Whenever gaseous fumes like these are going up, and again it is coming back as contaminated rains, these also end up going into the water body. So these are different indirect sources which are polluting the water. And then comes the soil pollution, which is the most drastic, I would say, because we are in usually in direct touch with soil. And the 30% of soil that is present on this planet, whole lot of it has already been contaminated by human beings. May it be agricultural practices or malpractices, may it be different industrial waste or whatever it is. Every day, for example, even if we consider ourselves, every day we are disposing the plastic, these trash bags into the environment. So we are constantly adding up these. Every time we are drinking a Coca-Cola or a cold drinks or even a cup of tea, we know that the paper teacups also contain plastic linings inside. So in every way possible, in every possible way, we are every day contaminating or polluting the environment, and we have been doing so happily for since so many ages. Especially if we talk about the industrial revolution, definitely after that. So these all together is heavily, and it has been heavily polluting the environment so far. And who are the end sufferers? Definitely different organisms, fishes, aquatic life forms, terrestrial life forms, plants, animals, including us as well. So for example, if we talk about the different various effects of pollution, talking about air pollution, it affects our normal lung functioning. It leads to respiratory disorders like asthma, and that is nothing new for us now. We all are very much aware of this cough and wheezing, itching of eyes, especially in heavily polluted cities, in esophageal tract, eyes, headache, and neurological disorders have been reported to increase over the last many years, and as well as it negatively impacts the reproductive and immune system of human beings. And of course, cancer is always there associated with this kind of unhealthy environment. Talking about water pollution, it causes different waterborne pathogenic diseases like typhoid, amoebiasis, giardiasis, hookworm diseases, and so many. It's a huge list if I start counting. It leads to rashes, ache in the ears, pink eye formation, hepatitis, encephalitis, gastroenteritis. So many diseases are waterborne. Liver and kidney damage. It can lead to DNA damage as well as Parkinson's disease due to heavy water. This high molecular weight or heavy metal pollution in the water system. Sorry, and from soil pollution, the soil, different anthropogenic pollutants in the soil, they affect the brain, hindering the normal mental development of a child. It definitely leads to cancer as well as damage to the central nervous system, different organs like kidney, headache, fatigue. So these are the different. So it's basically not possible to enumerate, all the ill effects that these different sources of pollution cause to human beings. This is just I would say, snapshot of some, just very few of these, few of the effects that these pollutants like carbon monoxide, heavy metals, oxides of sulfur and nitrogen, particulate matter, ozone, VOX or volatile organic compounds, parasites, pesticides, organic pollutants. So all together, they are basically not leaving any part of the human being which is unaffected by these pollutants. So this is high time we should always keep this image at least in our brain whenever we are polluting the environment. Now talking about pollutants, especially there are a lot of different pollutants as we have seen here also, but specifically my discussion now would move more towards organic pollutants because I'm primarily focused into this kind of pollutants. So uh, organic these pollutants have been classified into different four different categories. One among them are the organic pollutants. Apart from these, there are inorganic fertilizers, heavy metal pollutants, as well as radioactive isotopes, that is radioactive pollution. These organic pollutants are structurally most diverse and involved in many different deadly harmful effects, and we are mostly prone to this. We are mostly exposed to these organic pollutants as compared to the other forms. So almost every day we are getting exposed to in some or other way into these organic pollutants. So they have definitely certain deadly harmful effects for which we are concerned about these organic pollutants, and they need special attention to be dealt with. So what are these basically next to the glucosyl residue, as you can see here, the glucose structure, the chair conformation of glucose. So next to glucosyl residue, which is a natural ring, the benzene ring is the unit of chemical structure most widely spread in nature. So if you are not aware of this, this is very interesting that apart from next to glucose residue, benzene is the next residue which is most abundant and most widely spread in the nature. It is thermodynamically very stable, we know, because of the aromaticity that it has. And as the number of ring increases, for example, this is benzene. When there are two rings, we call it naphthalene. When there are three rings, we call them anthracene or phenanthrene. So as the ring keeps increasing, they become more and more stable and thus more and more persistent in the nature. So many of these pollutants are recalcitrant. Recalcitrant means the pollutants which stay or remain in the environment for a very, very long period of time. And upon bioactivation, for example, they can lead to formation of free radical. They can reactive. They can form reactive oxygen species. They can form oxybridges. They can form adducts. So in many ways, they are very much toxic and harmful for us. So once bioactivated, they exhibit more toxic and or can in many cases carcinogenic properties, that is they can cause cancer. So there are several types of aromatic nuclei or aromatic compounds in the nature, or anthropogenic compounds, that is man-made synthetic compounds. Among them, the two ones which are most important are and most dangerous are classified into two categories. One is endocrine disrupting chemical, the other is polycyclic aromatic hydrocarbons. So these are the two categories that I will gradually in my next slide I will discuss about next few slides. They are considered as most priority pollutants and by most of the environmental protection agencies around the world. For example, EPA or Environmental Protection Agency, USA, and several other countries. So what are these EDCs or endocrine disrupting chemicals? We know that in our body we have several endocrine glands which secrete the hormones and run and keep and helps in proper functioning of our body, of the system. There are certain chemicals which mimic those hormones and which mimic the endocrine system and can perturb or disturb the endocrine system. They can alter the function of the endocrine system and can cause adverse health effect in the organism. One very common example, you might have seen or heard that during pregnancy the doctors very strictly they ask the mother to stop drinking or avoid drinking water in plastic bottles. Rather, they should use either mud, clay pots or glass bottles, but strictly not uh, plastic bottles. The reason, because plastic bottles or whenever these PET bottles are synthesized or are manufactured, so these polymers usually use a lot of plasticizers, and many of those plasticizers they leach out. So when you keep water in the water bottle and that water bottle is getting exposed to air, sunlight, or heat or whatever it is, moisture, gradually the polymers are getting weakened, and these plasticizers they gradually leach and dissolve, get in touch with the water, and when we are drinking the water it is getting straight away to our gut, to our system. And if many of those plasticizers or phthalates, what we call them, or phthalate esters specifically, they act as endocrine disruptors, and uh, they have been found to change the gender or sex of fishes. It has not been tested yet in human, but it is speculated the same would happen to humans also. So they are so dangerous, they are so adverse that they can even change the gender of the fetus. So that's why or can perturb with the hormonal balance of the fetus. So that's something which is something we should be highly worried about. So these compounds which are classified as EDCs or endocrine disrupting chemicals are highly heterogeneous. That is, there are several kinds of these compounds. For example, synthetic chemicals used in various industrial solvents or lubricants. For example, polyhalogenated biphenyls or PHBs and dioxins. They are very frequently and very commonly used in industries dealing with solvents and lubricants. Bisphenol A, you might be aware of this name. Bisphenol A has been found to be uh, very toxic, and many plastic products, especially baby products, are now strictly they are eliminating bisphenol A from their composition. And plasticizers such as phthalates that I was just now talking about. Pesticides such as methoxychlor, chlorpyrifos, DDT. So these DDT also has been banned, we all know, but still we know that there are certain reports coming up now and then where it is still being practiced in some places. Fungicides such as vinclozolin, pharmaceutical agents, diethyl tilbestos or DES, non-ionic detergents, polyethoxylates. So these are certain compounds, certain class of compounds which can hamper or which can affect our endocrine system, and we should be worried about this and take care of this. These are some of the compounds, the structure, how these look like. uh, Phthalate that I was talking about is this one. I'm talking about phthalate because one of my research is also dealing with phthalate esters, which are endocrine disrupting chemicals. And apart from that, the next class of even more diverse class of pollutants are polycyclic aromatic hydrocarbons, also known as PAHs, which are ubiquitous pollutants. That is, they are found everywhere. These are mostly formed by incomplete combustion of coal, tar, oil, and gas, as well as from automobile exhaust and other organic substances like tobacco, smoked or charboiled food. So these are the common sources of pH molecules, that is polycyclic aromatic hydrocarbons. Apart from these, fossil fuels are also a major source of polycyclic aromatic hydrocarbons, and when fossil fuels are being burned, they these are also released into the environment, and we can get in touch with this very easily. Remember that even tobacco or smoked food also contains pH molecules. So the next time when you are going somewhere to have barbecue, be aware of this a little bit, especially the ones which are incomplete combustion, where incomplete combustion conversion is taking place. So various environmental protection agencies I was talking about have been considered or label them as priority pollutants. And as you can see here, this is the definition of a polycyclic aromatic hydrocarbon. That means two or more aromatic rings fused together. Remember, biphenyl is also polyaromatic, but we won't call it polycyclic aromatic because they are connected by a pi bond here. But when the rings are fused, as you can see here, so that the aromaticity can circulate, the electrons can circulate among the rings. So they are called polycyclic aromatic hydrocarbons, and they are far more stable. So these are some of the 16 priority pollutants which are very toxic for us. Some of them have been classified as carcinogenic for us as well. Among them, naphthalene and phenanthrene are the two ones which are most widely studied, because these are the simplest pH molecules with 2 and 3 ring structures, and they are quite often considered as model compounds to study the degradation of these kind of compounds in the environment. So now, talking about biodegradation of these kind of compounds, there are three different ways how these compounds can get uh, degraded. One is photolysis, that is by direct sunlight in the air. So quite often by intense when the sunlight is intense, it can cause photolysis of these compounds, especially the smaller ones. In water, there are different ways: photooxidation or chemical oxidation, and water and as well as soil and sediment also. So photooxidation and chemical oxidation can be implemented, are some of the ways of getting rid of these compounds. But among them, the most widely used or the most preferred one is biodegradation. So what is that? This is the utilization, that is decay or breakdown of an organic compound as sole source of carbon and energy by microorganisms. So whenever any microorganism is utilizing a carbon compound as its source of carbon and energy, and breaking it down to get nutrients or to get energy, that would be called as biodegradation. Uh, it can be a bacteria, it can be plant. In case of plants, especially, we give a separate name and we call it phytoremediation. So some compounds can be simply biotransformed. That is, they can change from one form to another form, and there are certain compounds which can be completely mineralized. That is, can be broken down into carbon dioxide, water, and these kind of byproducts, which go into the environment, back into the environment. This is most desirable when the compound is getting completely mineralized, because if a compound is getting biotransformed, there are chances sometimes that the transformed compound might also have some different kind of toxicity. In certain cases, these can even be more toxic than the parent compound. So, but anyway, there are two different fates of these aromatic hydrocarbons. Now, biotransformation or biodegradation can either be bacterial, algal, or fungal, and this can so far has been considered as the best approach. It is also quite often referred to as green chemistry. So, best approach till date, and has become an intensive area of research, especially in last I would say 30 to 40 years. Now, talking about biodegradation, the removal of pollutants from the environment by physicochemical process, in general, a slow and unpredictable way of counteracting pollution. So these physicochemical processes are expensive, sometimes a little bit harsh to the environment itself, and unpredictable, and how and when this will happen. So the main strategy to fight pollution is the use of manipulate the detoxification abilities of living microorganisms. So this is very important here, because living organisms or microbes per se, many of microbes have their own detoxification abilities. They can degrade the compounds, but sometimes the wild type organisms are not good enough to decontaminate the environment to an extent to which we have already contaminated it. So in certain cases we need to take the potential of these organisms and do certain manipulations with what we refer to as genetic engineering or molecular biology, that we will also discuss about, to get rid of these compounds. And by expressing different catabolic pathways, microorganisms can degrade a wide array of aromatic compounds. So many compounds have been studied so far. And however, these xenobiotic compounds or man-made or synthetic compounds have been in contact with the microbiota only for about 100 years. So the microbes, they didn't get, so far they haven't got a very, we all know that evolution has been occurring since million of years, right? And microbes also have been there since the very beginning, and they have been evolving a lot. But talking about you know degradation of xenobiotic compounds, these xenobiotic compounds have come into the nature not more than 100 or 150 years back. So the microbes have hardly got a little bit more than 100 years to adapt to these compounds and to evolve, to degrade these kind of compounds, right? So sometimes that is the reason which limit their use as potential degradation tools in the environment. But again, using different technological approaches, using designing different novel strategies, we can find out ways, to take help from these microbes and combat these pollutants. This is a overall strategy of how we search for bacteria, especially I'm talking about bacteria here, not fungi or algae. So to find out how, where and how many bacteria are present which can degrade these kind of compounds. So the common strategy that we usually use is we collect different soil samples, especially from contaminated places where we might think that the inherent bacteria, the local microbiota, might already be exposed to certain chemicals, certain pollutants. So we collect the samples. It can be water sample, soil or sediment sample, and then we put them into enrichment cultures. So what are enrichment cultures? These cultures are media, and in most cases these are usually carbon free media, where we put the substrate that has to be degraded as the carbon source. So we put a media here, we do not give any other carbon source apart from the toxic compound. So now the bacteria has two choice. Either it will if it has the potential, it will try to degrade the compound, break down the compound and derive carbon out of it, or it will fail to grow. So that is the only way to uh, escape or to survive. So now what happens is, we continue this enrichment for several cycles over a period of 1 month, 2 months, 3 months, based on, depending on the compound, the robustness of the compound or the persistence of the compound that we are dealing with. And then finally we end up having certain bacteria. Maybe it can be single bacterial cells or some consortia, that is mixed bacterial culture, which become adapted and which are known to be capable of degrading this that particular compound. And all those bacteria which fail to degrade or fail to survive, they will basically get eliminated over due course of time. And then we plate the culture, and we take the isolates. And for example, here we found three different isolates, so we will put these three differently in cultures, again with the same substrate, to see whether they have been growing as a mixed consortia or any one of them has the potential to degrade itself. So if we find that, say for example, here out of the three colonies, at least two cultures failed to grow and only one of them showed growth, so we then plate them further and we identified that yes, this is the potential degrader which was growing here, and maybe the other two organisms were depending on the metabolites degraded by this compound, this strain. So now we take the culture as our single isolate or degrader strain, and based on our research goals, we then can move towards degradation pathway analysis, which comprise of biochemical, analytical, molecular studies, we can go for genomic analysis, we can go for molecular biology techniques, recombinant DNA technologies like that. So talking about the techniques that follows after the bacteria has been isolated, there are primarily three different ways. One is biochemical analysis, where we perform solvent extraction and certain analytical tools like TLC or thin layer chromatography, HPLC, gas chromatography, mass spectroscopy, GCMS, for purification of the compounds, as you can see here. And in many cases, some of the compounds, some of the intermediates that we come or that we isolate, we need to understand the structure of the compounds. In such cases, we go for nuclear magnetic resonance or NMR structure, and then based on the NMR spectra or the NMR spectra, we basically try to understand the structure of the intermediate or the metabolite. So these are certain techniques, very frequently used, certain analytical techniques used in biodegradation studies. And see, here I have written biodegradation, a blend of scientific fields. Why blend? You will see gradually. So first we talked about this analytical techniques. Initially, it was pure microbiology that we have been doing. Now we have moved towards analytical tools. Apart from analytical tools, we can take the bacterial cell, isolate its genomic DNA, or if it has plasmid, maybe plasmid DNA, and then can sequence the entire genome. These days, getting a genome sequence is not a big deal. So we can get it sequenced, and we can do certain bioinformatics analysis. We can do certain mathematical or statistical calculations upon them, and then we can come up with the degradation, how the degradation might occur in the organism, what are the genes responsible, what are the proteins responsible, what are the regulatory factors, what are the regulatory, where are the promoter sites, how the operands are being regulated. So many questions can be answered once we have this genome sequence. Or else we can straight away go towards molecular biology. If we have the genome sequence, or at least if we are aware of the sequence of the gene or the operand, it helps. So what we can do here is we take the wild type cell that we had isolated from the environment, we take its chromosome, and we know that somewhere in the chromosome there is probably one gene or certain genes which are of our interest, that are actually involved in the degradation study. So we first try to assess where in the chromosome that particular gene lies, whether it is in the chromosome or whether it is plasmid bond, whatever. So say this particular gene, this blue one that you can see here, was somewhere in the chromosome that we are interested in. So what would be our next strategy? Our next strategy will be to amplify that gene, to cut that particular gene and amplify the gene exactly. So we first amplify the gene using PCR. We put certain restriction sites in the primer, so that the amplified gene will have certain restriction sites flanking the gene site, and then we can digest them. And what we will do after that, we will take another plasmid vector which was isolated from some other organism, so a very standard plasmid vector, and we will digest this vector with the same restriction endonucleases or restriction enzymes which we used here, so cutter 1, cutter 2, so that it gets opened up and this fragment is lost. So now what we do, we take this gene, because you might be aware of this, that this gene now contains the sticky ends as same as the stick compatible to this particular plasmid vector. So we will just bring it, we will ligate it here, and after ligation, this plasmid vector will now turn into a recombinant plasmid, right, as you can see here. So the plasmid containing our gene of interest. Now we will take a cell which is suitable for expression of this particular gene. We will make it competent. For example, in most cases it is usual in E. coli. So we will make the cell competent, and we will perform transformations. What is transformation? We will put this plasmid, we will insert into the cell, either by chemical transformation or electroporation, whatever we wish. And then we are getting actually our recombinant cell, or recombinant E. coli, which contains this particular gene which was of our interest. And now we will try to manipulate this gene. We will try to we can try to get the crystal structure of this protein. We can overexpress it, and then try to perform remediation studies. And so many things can be done once we have this recombinant cell with us, or the recombinant gene with us. So now biodegradation is basically a blend of scientific fields. Just in last few slides, you have seen that we have been using analytic microbiology, analytical tools, informatics, that is computer mathematics, and well as genetic engineering or molecular biology. So basically, biodegradation, or people who are working in the field of biodegradation, are working in interdisciplinary research. Why and how? It contains microbiology, definitely. It contains a whole lot of chemistry, because you are dealing with pathway intermediates. You are characterizing molecules, and at the same time, these days nothing can be done without informatics or bioinformatics. So definitely you have to have some exposure to information technology or bioinformatics, as well as, in certain cases, not always I would say, but in certain cases, mathematics. So, and yeah, don't be scared that you have to know everything. I'm not saying that one particular person should be expert in all these fields. What I mean here is, for a good biodegradation approach, to develop a good biodegradation approach, or for a complete in-depth study of biodegradation, you need to deal with the problem using microbiological techniques, chemical techniques, taking the help of information technology, and if necessary, statistics or mathematics, and then you will come up with certain good results, definitely. So these are certain applications of biodegradation, because at the very first place the question comes is, why we are wasting so much of money, research, energy, time, and everything. So definitely there must be certain applications of biodegradation, because the microorganisms can be used for decontamination of soils. It can be used in situ in remediation of contaminated groundwater. We can get rid of toxic heavy metals. We can develop bioreactors for biocatalysis using biofilms and the purified enzymes. And these kind of enzymes can have other industrial applications, for example in food industry, paper and pulp, textile, like that, or in synthesis of synthons. So these are basically drug intermediates from which different drugs can be designed or synthesized. And genetic information can also be used for constructing bioreporter strains. So they are very hot topic of hot research these days, constructing bioreporters. Now, talking about what I exactly have been doing so far, is that my research actually deals with both experimental as well as computational analysis. So among experimental, what I usually do is I isolate these kind of organopollutant degraders, that is bacteria, from contaminated environmental samples, try to understand the degradation pathway by the analytical techniques that I was just now talking about, and then basically then dwell towards the genome or sequence of those organisms, and try to understand the pathways from there, how it has evolved, the enzymes, how those enzymes have evolved, and so on. And another aspect of my research is the computational one, where I try to understand how these enzymes have evolved. And uh, because I deal with full lot of enzymes, one of my interest also is construction of online databases and servers that users can access and do their analysis there. So now I will not go into exactly what my research is in depth, because that is not very important here. What I would like to rather show you are certain images and certain data, to show how this kind of research, what can you expect out of this kind of research, and what kind of data basically we end up with. So this is basically a degradation pathway of naphthalene and phenanthrene by one of my strains. I will show the reference at the very end. So this is these are the structures that are within parentheses are the structures that we couldn't identify, and the compounds which are written this way are the intermediates that we had identified. And these are based on speculations that if this compound is forming from this, definitely it has to go from these intermediates. And you can see certain cross symbols here. That means these are dead end products. That is, once this is produced, it cannot go this way. And how we are knowing that this is produced? Because we have some certain cases are knocking down certain genes, or we are expressing only this particular gene, cloning and expressing these genes, and we are ending up of these metabolites. So we know that yeah, how these enzymes are working. So this is the result of different gene knockout experiments, or certain cloning experiments, and certain growth kinetic studies. And a lot of enzyme activities are measured to understand the role of each pathway enzymes. The small purple names that you can see here, maybe you cannot read, I don't know, but these were identified by a lot of enzymatic and spectrophotometric analysis. And the intermediates were identified based on HPLC in certain cases, GC gas chromatography, that is GC mass, or NMR studies. Apart from this, the degradation pathways. Yeah. Now, one thing I would like to mention here, this is encircled because apart from studying the entire pathways, the pathway, one of my prime research interests is the first gene, which is called the ring hydroxylating gene on risky oxygenase gene, because uh, this is the first enzyme which is making the otherwise inert or very stable, this hydrocarbon a little bit hydrophobic, so that other downstream enzymes can come and act upon this. So this is the most important enzyme, and it has a wonderful role because it is basically making a very stable compound prone to further attack. So this is called risky oxygenase, which is basically a multi electron transport. It is a multimeric protein having certain electron transport components. For example, the reductase, which gets reduced by NADH, and once reduced, as you can see the electron, it passes on the electron to ferredoxin goes and gives the electron to this terminal oxygen, and once reduced, this terminal oxygen basically does the hydroxylation, or rather dihydroxylation. So it was from one of my strains NJ, where I had cloned the entire gene fragment into one plasmid vector, and then from the recombinant strain we had seen the biotransformation and characterized the different intermediates uh, which these genes. So these are the alpha and beta subunit. This is the small one is the ferredoxin, and this is the reductase, as you can see here. And this was a transposon that we by accidentally came here. Anyway, so what I mean here is, by using so many analyses, we finally came up with this entire pathway. So a lot of spectrophotometric analysis, a lot of different analytical tools like HPLC, GC, NMR, as well as cloning experiments finally gave us the answer to this particular problem, that through which the degradation was going on. So further working on this particular enzyme, I further worked on a lot of quite few years after that. I worked with on the evolution of that particular enzyme. This is the catalytic pocket, where you can see here. I'm not going again into details. These are phylogenetic analysis. So what I finally found that I would like to share with you, as I mentioned, that these kind of pollutants came into environment not more than 100 or 150 years. So I wanted to understand that if this enzyme has evolved to degrade these compounds, what was the actual compound, or what was the actual source or protein from which these enzymes have evolved. So while doing that, and that is published also, we finally came to know, after a rigorous study of like maybe more than 2 years, that there was certain proteins we call BET-V1 like superfamily proteins, and it was a pollen allergen. It used to be a pollen allergen, say maybe many million years back in certain plants. Those proteins or those enzymes gradually evolved towards, with the catalytic pocket which can accommodate these kind of highly hydrophobic compounds and aromatic hydrocarbon compounds, and can degrade them. So that was the study that we found out of our evolutionary studies. And this is the proposed pathway that we usually but that we have understood, that these kind of degradation pathways, or for example, if if we are talking about microbes, they will contain intermediate pathway which will basically lead towards the TCA cycle. So this is the TCA cycle. All the pathways must go and merge here to get degraded completely, utilizing the carbon and energy. So this TCA cycle, or the central or intermediate pathway, central pathway will get connected to different pathways like intermediate pathway and peripheral pathways. In certain cases, if certain enzyme is missing, some other enzyme will come and do the work. It will transform this to this. So this is how evolution of metabolic pathways take place. These are modular in nature. For example, if this is a pathway, one peripheral pathway can come and connect here in one organism. In another organism, the pathway can come and connect from this intermediate and go this way. For example, we have seen around during glycolysis and different carbon synthesis, carbohydrate metabolism. This is how it works also there. So in case of aromatic hydrocarbon degradation or xenobiotic degradation also, the pathways have evolved this way. And there are certain enzymes which go through alternate pathways if the original enzyme is missing. And these are certain mathematical derivations. As I just now mentioned, that I was trying to figure out that what was the initial stage of this enzyme. So to prove that, I had to use set theory in mathematics, and then we named it the traceback method, an approach to look back like many thousand or million years back, how these enzymes have evolved over the years and have become these pH degraders these days. And because during this all this analysis, I ended up with a lot of data, and I had to gather the data from various sources. I felt that why not make a database itself, so that fellow researchers like me who are particularly working in this particular enzyme system, they can get benefited. And that was in 2014 when we published a online database which is available online. Anybody can go and access, which is basically a database as well as a prediction tool. For example, if you have a chemical structure, you can go to the database and you can draw the structure basically in the database in under analysis module, and the database will give you possible risky oxygenase which can degrade the compound. Or else, if you have the gene sequence or the protein sequence, you can put that sequence here, and it will give the structure of the compounds which the particular protein might degrade. So this is how the database works. So these are again certain results based on functional prediction based on that data. So this data is that you can see here, we published much later in collaboration with Thailand and India. So this was completely based on the output of this data by using this database after a few years. So this was published in 2014, and this data was published in 2017. Unfortunately, I forgot to mention the citations here. So this is how we now predict different oxygenases based on this database, that what can be their function, and accordingly we design our experiments. And then based on our theoretical knowledge, based on the database, we now design experiments and go into degradation studies. Gradually, when I moved to Japan, I got interested, because until now I've been working with mesophilic enzymes, that is from normal soil bacteria. So now I focus my research towards searching for thermophilic homologs, because we know that thermozymes are very important. They are very good for various industrial processes. They are resistant to proteolysis, different chemical denaturants, detergents, organic solvents, so on. So while doing that, searching for novel thermophilic homologs of risky oxygenases, I came across a bacteria. It is called Thermus oshimi GL2, which was actually isolated by Professor Brian Hedlund. He is is a good friend of mine. He is in this University of Nevada in United States. So he gifted me this strain, and in one of the strains, one of the plasmids, or rather we call it megaplasmid of this strain, I found a very interesting pathway which was very unique, not found in any other bacteria, but only in very few Thermus genus. So I've further explored, like next 2 to 3 years, not exactly 3 years, next 4 years, I worked on this particular plasmid, this particular pathway, and we came up with a unique cluster of genes and a very unique pathway of lignin, degradation of lignin aromatic hydrocarbons. That is, we call them lignoaromatics. As you can see this graph, you can see the thermostability. So this is the temperature, and you can see the enzyme, the optimum temperature of the enzyme is 70 degrees, whereas at 80 degree it is almost 90% active, and even at temperature as high as 90 degree, it still is showing 40% of its activity. So you can understand how thermostable these enzymes are. So we came across with a couple of enzymes. One is the risky, and another was the decarboxylase. We tried to knock down the gene, and we found that the recombinant, that is the knockout mutant, was not showing the activity. So we finally proved that yes, these two genes, especially it is this gene and this gene, these two genes we had found to be thermostable. Out of them, I couldn't knock out this because it was a multi-component enzyme, but successfully we knocked out this gene and proved that yes, it was a very functional enzyme, and it is very industrially very important enzyme. So currently we are working on this. So this was all about what, very briefly I would say, what I've been doing in last 20 years. I tried to complete that in 4 or 5 slides. So this is my current research team. They are my research students, PhD scholars, and some master students also. I don't have image of very recent students of mine, and these are certain sampling images. We go through a lot of adventure. Sometimes our car gets stuck and we have to push it. Sometimes we go to places where cars cannot reach, so we need to sit on trucks and or walk certain miles to go and do sampling. And these are the publications, not all, but some of my publications, the data of which I have shown here in my previous slides, the previous slides. And these are certain websites which I have used to get certain images. And yeah, finally, the presentation cannot end by acknowledging my funding agencies, beginning from my PhD days, that is CSIR, my postdoc in India, where I got fund from DST, Department of Science and Technology. In Japan, initially I was as a JSPS postdoctoral fellow, funded by JSPS, and then I became a faculty member of University of Tokyo. During my PhD, I did completed my PhD in Bose Institute, which is a DST institute, where I was a research fellow, JRF and then SRF. So I acknowledge them. They are my is my PhD guide, Professor Tapan Datta, and Professor Hideyoki Nojiri, who was my collaborated in University of Tokyo, and he was the person who actually invited me to Japan and encouraged me to apply for this. And definitely my university, CHARUSAT, Charuta University of Science and Technology, for allowing me to continue my research and for allowing me to present the talk today. And not last but not the least, Gopal sir, for kindly inviting me and giving me this opportunity to share whatever I have experienced over the last couple of
बहुत
decades.
अच्छा शेयर